Okay, thank you first for the invitation to talk about eDNA and the project here. I will start a bit about the background and everything, and I will get back to a few results from the COREu project in the end of the talk. But first, just a quick introduction to who we are. NINA, the Norwegian Institute for Nature Research. So we are an independent private research institute in Norway. We do mostly applied research. We are more than 350 employees all over Norway, and more than 80% of the scientific staff has a PhD. Most of the work we do is inside Norway, but we also have quite a lot of collaborations outside, both in Europe and on other continents. We are sitting in Trondheim at the moment in central Norway. We are sitting in this beautiful building that you see here. And we have offices also in other parts of Norway. NINA typically do applied research where there are conflicts, for example, between humans and farmed animals, or hydropower, for example. So we have covering, or are covering, most of the biomes in Norway. At NINA, we have the Center for Biodiversity Genetics that was launched just a few years ago. We have a large technical staff of 9 permanent lab engineers that do most of our lab work for our projects. So in COREu, how do we use eDNA? Well, all these nature-based industries and activities, they have an impact or may have an impact on nature. And in this project, we are trying to make a baseline. What do we find today before we actually start changing stuff, before we start building? eDNA is a new and innovative method for assessing biodiversity. And in COREu, we will use eDNA to assess marine biodiversity in the Gulf of Kavala in Greece. So the design has already been set and samples have been collected. So you have this gradient from the shore outwards to the proposed storage reservoir. There are 14 stations where water and sediments already have been sampled, and they have been sampled 3 times a year to cover seasonal variation. The eDNA will be extracted here at the NINAGen in the lab at NINA, and we'll store all these samples in our biobank for the future. So if there will be a leak in the future, anything happens, we have the eDNA samples in the freezers. We can always go back and redo analyses, and we can run new analyses at any time in the future. We can also select different samples, of course. So this is a cost-effective way of having a biodiversity analysis. We're going to run 2 different primers that we already have tested in the lab. One that targets the foraminifers, and a more broader primers that are specifically targeting invertebrates, but also one that gets fish and other organisms as well. So what is eDNA? I'm going to talk a bit about what it is in this talk. I'm going to talk about quite a few of the projects that we run at NINA in other biomes. So eDNA is called conservation in a cup of water. And the idea is that in a water sample, we have small remains of DNA floating around. And by harvesting the eDNA in the water, we can find out what species are actually in these water bodies. Very easily, we take a small water sample and we filter it through a small filter to preserve the DNA. And then we get into the lab and analyze the data. eDNA can be anything. It's all the materials that we can use to extract DNA. So it can be water that we use in this project, or sediments. It also can also be air. Just sampling air and filtering air can get eDNA. And we also use soil samples for bacteria and plants, etc. If you go really down in the sediments in lake, for example, you can also get old sediment. There's also a form of eDNA, and we can find traces of mammoths and historic wildlife. So eDNA is the sample itself. It's the soil, it's the water, the air, and we have 2 very different methods for assessing diversity. I think it's a bit important to have that in mind when we look at the results. So the first one is a method where we detect single species only. So that means we have genetic primers. We use genetic methods where we can find one species at a time. This is often used to find rare red-listed species or unwanted invasive species. And both of these groups are very relevant for the management. The second method, called meta-barcoding, then we want to find as many species as possible with one analysis. We often use these to look at diversity, to look at ecological condition, and we can use these for environmental impact assessments. So with the first method, we run qPCRs or digital PCRs. That's the most common methods. There are also others. But what we see from the results is basically what you see from these graphs. We never see actually the DNA code itself. We see that we have a species. We can also measure the concentrations of our species DNA in the water. It's very simple. It's quick. We can do this in a few hours in the lab. And it's quite cheap relative to the other method. The other method, we actually get DNA strings. So we have the true code. And what we do need is a reference database. So all of these lines now are different strings that we can get from the water samples. And you can see there are some mutations. The genetic code is a bit different from the first. It's 3 sequences in the middle that are a bit different. And then the last part are different again. So comparing these sequences to reference, we can see, okay, the first ones are actually from pike, the fish, the northern pike. The middle ones are from roach, for example. And the last ones are from trout. And that means we can look at fish species. We can also look at how many DNA copies do we find from each species. And we can make these relative distribution in the form of a pie chart. It's quite complex to get to these sequences. And the lab work is much more heavy. We need much more bioinformatics to clean all these sequences and get it compared to the reference library and then get the species list in the end. So just looking at the data we'll get from these analyses, we have been using the traditional Novosec for many years. We have 800 million DNA copies. That's what we get from one analysis. And now the new machine is just being launched, and we're using that now in Oslo, in Norway. And it's 1.5 billion DNA copies per analysis. So there's a lot of data being generated by these analyses. This is also why the second method is much more complex. But why should we use eDNA? Well, taxonomists are red listed. I put it in quote mark, but it's really true. I also put in a photo of Charles Darwin here. We are losing a lot of the taxonomical experts in the world. Most of the museums are losing their experts, and new ones are not being trained. And being trained at these different taxonomical groups really takes a lifetime to learn all of this. When we have an expert, they're often expert on one specific group of the diversity. So we don't have one expert that can know all the different groups. So we need a lot of taxonomic experts to cover the full diversity. With eDNA, we can also get genetic variation within species. I'll get back to that a bit later. But that means in addition to species, we can go below the species level and look at populations. It's very cost effective compared to having traditional morphologists analyzing one by one individual. We can do many at the same time. And it's also simple collection methods, sampling methods. So we have used this also quite a lot in citizen science, where the audience can actually contribute samples. And what can we learn from eDNA? Well, species presence is the obvious thing. We can say if a species is there or not. This is very often used then to have threatened species or alien invasive species. We can look at distributions. If we have samples from many rivers or many parts of the ocean, we can compare and see where are these different species. And then we look at species diversity. How many species do we find? And we can do community analysis. Do we have the same species communities in different places, or do we have different species communities? What we cannot do with eDNA is to assess number of individuals. We can assess the concentration of DNA per liter of water that we filter. But if that is many, many small fish or a few big fish, we cannot tell. So the size distribution, if there are small individuals or large individuals, we cannot tell. If there are old or young individuals, we cannot tell. And if it's females or males, we cannot tell that either at the moment. We are using now, instead of DNA, we are using RNA to try to mitigate some of these lacking stuff. And maybe in the future we can say something about age and sex, for example, from water samples. But at the moment we cannot assess these factors. At NINA we use eDNA across all environments. We are doing quite a lot in the terrestrial environment. We do most of our work in freshwater, and then we also do quite some work in the marine environment. We have developed our own kit, so we can send these out to citizen scientists. We also have the management doing quite a lot of the collection in Norway. So we send out the kits, the management can do the sampling themselves and send the kits back for analysis to us. We also have a small drone that we made. So basically this can drive on smaller lakes and even in the marine environment and collect samples over a larger area than representing water over, a complete lake even. We also work with marine plankton samples where we assess salmon lice, one of the major problems with fish farming in Norway. And we have built equipment to be able to collect these large-scale plankton samples. We're going to jump to these two different methods that I talked about. We're going to start with this single species method where we use qPCR and ddPCR to find specific species. And I'm going to start with a freshwater mussel, the freshwater pearl mussel. This is an endangered species in all over Europe, and Norway actually has between 40 and 50% of all the mussels in Europe are found in Norway. And that means that we have a special responsibility to preserve and take care of the pearl mussel. It's quite hard to find. It can be dug into the substrate in the bottom of the rivers, and you need to walk around with these binoculars, basically just to find it. And that means eDNA is a very nice method. You can take the water samples and we can assess, do we have the freshwater mussel here or not? And this is actually the species that in NINA we have done the most number of analyses. So we have thousands of samples for more than 1,000 rivers in Norway analyzed for this single species. We also used it to do some assessments for how far DNA is transported in rivers. In this example, we have river distance on the x-axis, and we have a large aggregation of mussels. So the dark gray bars here, that's mussel populations. You can see we have more than 900 mussel in one single site here. So this is a lake with a river with lots and lots of mussels. The eDNA concentrations are the circles with the concentrations on the left. And we've been here in different times of the year with May in blue, June in red, and then August in yellow. And you see the time of the year really strongly affects how much eDNA we find in the water. Then we have assessed it just beside the population, but then also 2 kilometers downstream. And although there are very, very few mussels, we still find almost the same eDNA signal in every month. So this means that eDNA is really heavily transported in rivers. And that means we can sample a few localities in a river to assess really long stretches of the river. Another red-listed species are the great-crested newt and smooth newt amphibians. These are not in rivers, but in ponds, a bit more difficult to assess abundance. But again, we want to know if these red-listed species are there or not. And here we also try to assess actually population sizes just using water samples. So we have these different ponds on the y-axis. Now we have the concentrations on the x-axis below. And you can see some of these ponds have no amphibians at all. And some of them have very, very high concentrations of DNA in these ponds of the two different species. So here we try to assess both the presence, but also the amount of amphibians in different lakes. And this is all just by filtering water. In the same project where the amphibians, we actually tested our genetic marker for chytrid fungus. And we detected this fungi in Norway for the first time using eDNA. It's never been found before. And we just happened to test the genetic marker in Norway, and we found it in 5 localities in southern Norway back in 2017. So this is actually the first detection in Norway, and it was discovered just by using water samples. So the chytrid fungi is a well-known invasive species And the disease is known for having degraded or destroyed more than 600 populations of amphibians around the world. So this is quite an important invasive species to monitor. Yes. We're going to jump to another invasive species, Gyrodactylus salaris. This is a fish parasite that is specific on Atlantic salmon. And in Norway, we have lots of Atlantic salmon as well, one of the largest population also there in the Atlantic. And this parasite was introduced from the Baltic Sea. So the Atlantic salmon and the Baltic Sea are resistant, where the Atlantic salmon on the other side of Norway and Sweden in the Atlantic are non-resistant. So in Norway, we had this now in more than 50 rivers. We have been actually treating the rivers with chemicals to kill off the parasites. In all these yellow circles, we have actually already treated salmon, treated the rivers to eradicate the parasite. And then we're left with a few regions. It's still a few rivers in the Oslo area, and very close to Trondheim, we have the river on the right, Driva River. And here we actually put up a barrier to stop the salmon. When it enters from the ocean, it enters the river. We put a barrier here on the map here. The ocean is on the left side. The salmon would come up to the barrier and be stopped. So that means all the way upstream, the salmon will be actually, after a few years, it will disappear, and then the parasite will disappear. So here we have used eDNA. We are now looking at eDNA concentrations on the left axis. We are looking at the river distance on the x-axis, and the barrier is the vertical line. So below the barrier is towards the sea on the left, on the right is above the barrier and upstream. So year by year, the barrier was erected here in 2017, and in the two blue colors, we have the salmon, the Atlantic salmon, the host species that we want to preserve in the upper axis. And we see when we put up the barrier, we can see the DNA being lost specifically from 19 then in dark blue and down to 2020 in this purple color. We can see that DNA is not only present as absent, we can also use the concentrations to see the amounts. For the parasite in the bottom, we can see from 18 in light blue, 2019 in dark blue, and down to 2020, it starts being almost away. So we have followed up this study now all the way to 2025. And even in 2024, we had a few cases of the parasite upstream, so it's still alive, but now it's gone. Using eDNA, we can keep control of the parasite and the host in the system. That was the freshwater. We can also move to the marine environment. Now we're looking at an invasive species that has been introduced from Japan. This is the sea water, or Japanese sea squirt, it's called. We call it sea vomit because of the way it looks. It looks like a bit of vomit on the seafloor. We found it in Norway for the first time in 2020 in these two sites marked in red. We went back in 2021, just half a year after the first observation by divers. All the circles here are actually eDNA samples. And we tested this by the genetic marker for the species. And we actually found it in both of these two places where it was observed by divers. And we did not detect it in any of the other water samples in gray. So that was good. But then we went back the year after and we expanded our study. We sampled all the way from the Swedish coast, along the Norwegian southern coast, and all the way to Trondheim up here where I'm sitting. And we can see that we detected these marine invasive species in quite a lot of places. So unfortunately, it has spread, and we have this invasive species in a lot of harbors in Norway. This study is done in a relatively few weeks of field work. We can do the lab work in a few weeks. So this is a quite rapid response, and it's a rapid method. If you want to repeat this with not using eDNA, you would have to put down divers at each site, start looking for this invasive species. So you have to use an ROV with a video camera to find it. So if you did not have eDNA and to do this, you would use months of field work, and that would cost quite a lot of money to be able to monitor this big area without eDNA. So here, again, it's quite cost effective and also quite time efficient to do this study. Okay, we're going to jump from the first method, where we do species by species, and we're going to adapt to biodiversity, where we use DNA metabarcoding to assess large communities at the same time. We're going to start on land, and we're going to look at insects. A few years ago, we realized that we are losing a lot of insects in both Europe and the rest of the world. The Guardian here puts out a very important study from Germany, where they have measured biomass, daily biomass, in a forest in Germany, and you can see a very large reduction. And it seemed like you have like a 30% decrease of the insect biomass in this period from 1990 to 2015. And we realized in Norway that we don't have a proper monitoring system in place to be able to detect such trends in insect diversity or even biomass. So in Norway, we started already in 2018 with testing. How do we do these? Should we use genetics? Should we use morphology? What kind of field sample methods do we use? So we have been developing this method. We're using a malaise tent. This is a trap where the insects fly in on the side. It hits a wall, and then it moves upward towards the light. And at the end, it crawls and crawls up to this point, and it will end up in this bottle. One such bottle can have thousands of species, but at least thousands of individuals. So you get all of these tiny, tiny insects. And to do this by morphology and get it down to species, you would have to do individual by individual and assess the species. And this is extremely time consuming. So basically, this is one of our colleagues here. It's sitting by the stereoscope, trying to determine these species. In the lab, we can do this quicker, sample by sample. And we had Menon Economics, a company here in Norway that tried to do a socioeconomic analysis of these. And if we do a full-scale insect monitoring in Norway, it will cost 40 million euros per year. Using DNA, we can lower that to 2 million per year. So the contrast of using morphology or DNA here is really, really large. And that is all because of the really huge time cost sitting and doing one by one individual and determining the species of these insects. So far, we're now covering all of Norway. We have actually detected 27,000 species of insects in Norway since the full-scale monitoring study started in 2020. But we only have 19,000 registered based on morphology. And we do think that we have more than 30,000 species in Norway. So that means even with a group like insects, where we should know the diversity, these are all flying insects. It's not these tiny insects that we find in the soil or anything. These are all flying insects that enters the trap that we use. And we don't know 1/3 of the diversity in Norway is completely unknown. So that means there is a lot to learn using genetics on these large-scale data sets. In addition to species, I mentioned this is one of the first slides, we can also go below species and look at populations. So in these two examples, I have put in two different species. So one graph is one species, and the colors are different genetic variants within the species. So this means that this is a cockroach. We have these purple genotypes or different genetic variants in southern part of Norway, and it turns to this green one, and we have the blue one where it's a bit higher altitude. So this is basically a bonus of using the metabarcoding method. We can assess the species, but we can also assess each different species. On the right side is the beetle. Some of the same pattern here. We have a reddish haplotype or genetic variant in the Oslo area. Going further north, we get a new genetic variant. So that's something extra we can do using genetics. We are jumping from the terrestrial system, and we're going to jump to the aquatic system. In NINA, we do quite a lot of work on hydropower. Hydropower is very big in Norway. A lot of our rivers have hydropower facilities. And we have quite a few years now, we had different hydropower centers like the Hydrosen, where we do research on hydropower. In these rivers, we try to assess the benthic invertebrates. We try to assess diversity, and again, we use only water samples in some studies, but we also use some other substrates. But in this slide, it's all water samples. What we can do from water samples, these are just comparing June and October. It's one single river, and we have different stations upstream the river to see how it changes. This is an order level of insects. So basically we have beetles, we have mayflies, we have all these different species, but you can see DNA is dominated by a single order, and that's the dipterans, that's the flies. If we look within flies and just look at the families, so just one order of insects, you can see the large number of families that we are assessing. And this is only water samples, and all these different families we can find with water samples. We can see some changes when we go upstream the river as well. Sorry, downstream the river. So this is what we do in hydropower rivers, but we also try to compare the methods. If we start with the left one, these are different orders of insects. And again, dipterans give the highest number of species. So this is actually the number of species per order of insects. The blue one are water samples. So this is just filtering eDNA and doing the analyses. The brown one is ethanol. This is actually what the morphologists use. They do a kick samples in the bottom substrate. They get a glass full of insects. And what we have done here is actually to filter the ethanol with those insects as an intermediate method. And then the purple one is morphology. So particularly for the dipterans, they're all these black small flies and mosquitoes. And we're talking about larvae here swimming in or living in the water. You can see the number of species what the morphologist can do with this group relative the number of species with water. And if you just look at all these different groups, the total, you can see in a water sample, we define on average per sample, we are approximately 60 species per sample. Doing this by classical methods using the taxonomical experts, we're down to maybe 15 species per sample. So we can find many of these groups that are really difficult to determine the species using morphology. Yes. We can also use this for the EU Water Frame Directive. This is an index that is heavily used across Europe in fresh water for assessing benthic diversity in rivers. So we can compare actually methods. Now again, the different colors are different methods. And the blue is the water samples. The brownish one is this intermediate method using genetics, and the purple is morphology. So we do get similar results using morphology and genetics for this EU Water Frame Index. We're going to move from rivers to lakes. These are all hydropower reservoirs, but now we are in still water in lakes. We are also moving from water samples to sediment samples. And remember now that in COREu, we are collecting both water samples and sediments. So I'm just showing some examples of the use of both of these sample types. So here we have tried to compare what is the effect of hydropower. So we have Trondheim in the middle of the map here, not being named, but you're trying to find these pairs of lakes where one is affected by hydropower and the other is not. So basically a control lake. And we've done these in pairs, quite relatively, at least geographically close to each other. In this particular study, instead of what we normally do is to investigate fish, and we do larger invertebrates, we are assessing the really, really tiny animals. So meiofauna, that is animals that are less than a millimeter, so they're really, really small. And the second here is protist, and this is single-celled organism. And they are even smaller. So can we assess diversity in this and assess the effects of hydropower just using sediment samples? That's the question. This is a bit complex one, so don't pay attention to everything. This is just the number of taxa we find in 2 different sampling periods in June and September. The lower one is actually looking at different phyla. This is on phyla level. What kind of DNA amounts, relative amounts, do we find here? For the meiofauna, the small invertebrates, we find more than 400 taxa or species. And for the single-celled, we find more than 2,000 taxa in the sediments in this study. What we wanted to assess was the effect of regulation. This is an ordination analysis. So basically, we take all the environmental variables that we have, and we try to assess these in single axes. So the axis here, the first axis is explaining most of the variance in all of the environmental variables. And the arrows is illustrating the most important factors. So what we found actually determined the variation for the myofauna on the left was regulation. Is there hydropower in that lake or not? So regulation, hydropower regulation really had one of the strongest effect on myofauna diversity, but also on the protist. You can see that regulation is one of the longest line and the strongest line here. So regulation definitely affects both the small invertebrates, but also the single-celled organisms on the right. The other one factor is lake size. So small lakes and large lakes have different communities. What we also assessed here, so basically, if you look at all the circles, they are the natural lakes, and all the triangles are the regulated lakes. So we could actually make a division here between the natural lakes on the inside and the regulated on the outside of this line. Same with the protists. We have the natural lake circles on the inside, and we have the regulated triangles on the outside. There really are a difference between regulated and non-regulated. So here we actually move even further, and we try to assess what functions do these organisms have. What do they do in the ecosystems? So we had groups of detrivores, mean they are breaking down material, for example. Filter feeders, that mean they are taking food by filtering the water. We have omnivores that are generalists, for example, are the littoral, that mean they're living close to the beach, not out in the water. We have predators that eat other of these small organisms. Suctor feeders mean that actually are benthic, they're sitting on stones, for example, to feed. What we do find is that, for example, omnivores, you can see that all the full circles are on the outside of this stippled line here. And this reflects the lines in the previous slide. So we can see that specifically for the myofauna, there are more omnivores in the regulated lakes and less in the non-regulated on the inside. There are also more suctor feeders. So that means not only diversity, but also the functions are different in regulated versus non-regulated lakes. Yes, we're going to jump in the end here to the marine environment, since that's where we're going to do the COREu study. And we have a program in Norway where we assess marine invasive species using both genetics and morphology. And again, we have done quite a few comparisons. In this project, we assess different sample types. We use plankton nets to assess plankton. We use the sediment in the bottom. We use water samples. And in the next slide, so I'm going to also show you these settlement plates, where you basically put out, these are plates made of plexiglass. We leave them out there for a few months, and then in the sea, different organisms will start to grow on these plates. So after a few months, we can take it back. We can basically just scrape off the organisms, and we can either use morphology or we can use genetics to assess the species that grow on these plates. On the first one, we have zooplankton. So that's basically net samples where we do plankton nets, get all the crustaceans in a small net. We homogenize them, we crush them together, and we use genetics and morphology. And in this graph, it's a bit difficult, but in the bottom, we have only the invasive species in color. In the upper one, we have all the species, whether they are invasive or not. So all the gray bars here are meaning they're not invasive, they're local species in Norway. For each box, we are comparing CON, which is the conventional morphology method, and the right bar is the eDNA. And each box is one phylum. So we have annelids, which is all these worms that you find on the bottom of the sea. Arthropods have all the invertebrates, the sea urchins, the sea stars, etc. And then we have all the algae and more plant phyla on the right side. So in a zooplankton samples, we will normally have a taxonomical expert on these crustaceans that are free swimming. So you can see for crustaceans that are in this phylum, there's quite a lot of species found by the morphological expert. But we can actually find even more species using genetics. But the morphology expert is not assessing many of the other phyla because he is an expert on the crustaceans, not the other groups. But as a bonus here, basically, from the zooplankton samples, we also find quite a lot of algae in the water and other groups. For the settlement plates here, pattern is relatively similar, but you can see there are other groups. This is one of the Rhodophyta. It's one of the algae that grows on these plates. You can see the morphology expert is more focused on these taxa than the arthropods. But we can find basically that using the same genetic markers on all sample types, we'll find species across these different groups. So what we end up with is that when you do conventional morphology using a species expert, all these different materials that you analyze are different because there are different taxonomical experts. So the zooplankton sample is only analyzed for crustaceans because it's a crustacean expert. The sediment samples are mostly, you have these worms, the oligochaetes that you find in the worms. The expert is assessing that. So you don't have any overlap in species communities between these different methods. In the eDNA samples on the bottom, you can see water samples and the settlement plates, the sediments and the zooplankton find quite a lot of the same samples because we have the same genetic markers across all the materials. So this is what we end up with. We do find more species and we can actually use fewer of the sample methods to find the same diversity as a conventional sample. But again, then, what you get from the conventional samples that I mentioned in the beginning, you can get the number of individual, you can have the sex of the individuals, and you can have size distribution, etc. Remember, we cannot get that from eDNA. We can only get species identity and some kind of relative abundance of DNA concentration. So let's jump to COREu. In Greece, we have the Gulf of Kavala, and the design of the project in COREu has been to sample from the shore and then outwards to the proposed site from the storage reservoir and even further. So station number 1 is closest to the shore and 14 then is furthest away. Just notice then that you're moving towards the deeper part of the Gulf. Then again, you are not that far from land again, from the shore when you go outwards. So I have a few slides of what we have done so far. And what we're looking at here are invertebrates. So basically, we're looking at the larger ones. So this includes the larger sea urchins, the sea stars. We have these oligochaetes on the bottom of the seafloor, etc. And this is all based on sediment samples. There are no water samples analyzed so far. So this is all small sediment samples from the bottom of the sea. We can see that in the shore, we have quite high diversity. And when you go out from the shore, it's going deeper and deeper, you get fewer species. We do have very few species here. This is actually the number of species per sample on average that we detect. So we do have less species than we expected. But that's what we have so far. We do see that we find fewer and fewer species when we go outwards. It seems to be in the middle where actually the CSS facility is going to be. It's actually a bit higher. I do suspect that this might be the deepest part then of the bay. Maybe there's a bit more shallow here. Richness on the top, and then we have 3 different indices assessing diversity. And what these indices do is that we use both the number of species, but we also assess how much DNA do we use? How much DNA do we find? Do we find a lot from one species, for example, and fewer of the other? So that's included in these different indices, but the patterns are quite similar. We find a bit higher at the shore when it's shallow, it decreases, and then we find more again when we get out in the end of the bay. So that's the inverts. We can also look at what are the phyla we find. The dark purple or dark blue that dominates most of the samples. We still have one sample by sample here on the x-axis. It's annelids. And that's these worms I'm talking about, these large hairy worms that lives in the sediment or on the benthic. It's quite expected to find in sediments. The light ones, the light purple, that is quite common in the shore, because this is the shore side, it's echinodermata, and that means basically sea stars, sea urchins, etc. And then, of course, they are also expected to be in shallower habitat. So it makes sense in that case. The other marker we have tested here are foraminifers. That's small calcareous organisms, very, very small. Again, we're looking at the different stations. So the numbers are the stations where 1 is closest to the shore and 14 then is furthest away from the shore. We have 3 different classes. So each bar, each axis here is looking at the relative DNA contribution from each of these classes. And you can see particularly the class, the dark gray or even black, that's the Tubotalamea class. It's also more common in the shallow parts, decreases, and then it increases again. So it shows similar patterns to what we find in the invertebrates. But then the other one, if you look at the other one, I'm going to show you just first a species. This is a specific species of foraminifers. This species, for example, is very common in the shallow parts, but not so common in the other samples at all. So this is probably one that thrives in more shallow environments. Another species like this one is not found at all almost in the shallow parts, but is more common when you get out in the deeper parts of the bay. So we can go all the way down to species level. But still, there are relatively few species, and we were expecting many more species. So we have to figure out where, how we can improve the analysis and get more species from these results. If you look at these three different classes, class by class, again, we're looking at the relative DNA abundance at different stations, but only from that class. You can see they have this increase in the middle from that. That's the dark one I showed you in the previous slide. But this one has a complete opposite pattern where you have really few in the deeper part. So yeah, this just shows that different classes, different groups are adapted to different habitats. So we need to go down on the lower taxonomic level to see these effects of for example, death. Yes. I think that's what I had. Just to summarize, so eDNA is really cost effective if you want to analyze many samples and you want to analyze a really broad taxonomical biodiversity and not these single groups that we used to do before, where we had taxonomical experts that knew these single groups, but maybe not all the groups. We can take the sample and we can store the DNA in our freezers for future analyses. So we get this increased taxonomic coverage. We can also look below species, as I showed from the insects, to look at the intraspecific genetic variation as well as species level. And then sampling is quite simple. So we also use this quite a lot at NINA for citizen science and even the management taking the samples themselves. Yes, thank you.